Good evening, everyone. I am Dr. Shiva Prasad. It gives me immense pleasure to welcome you all to this special lecture held in memory of Gregor Johann Mendel on his birthday anniversary. Mendel was born on July 20th, 1822, although his birthday is often cited as July 22nd, which is the date of his baptism. Today, we are here not just to honor Mendel, but also to celebrate his groundbreaking experiments with Gyan. Experiments completely changed how we understand heredity and laid the foundation for the field of genetics. Genetics has grown rapidly and is now used in many areas, from classical plant breeding to genomic selection and from medical diagnostic to gene therapy. This lecture has been organized by ShareGene WhatsApp group. We are a voluntary group dedicated to sharing knowledge in the field of genetics and plant breeding. Our group promotes continuous learning through discussions, scientific updates, quotes, basic concepts, short notes, MCQs, opinions. We regularly discuss about everything from basic principles to latest research and trends in science. If you are passionate about this field and want to go with us, please join our group. Let's all work together to share the genes of knowledge, because knowledge grows exponentially when it's shared. This lecture is not just tribute to Mendel's groundbreaking work, but also a moment for us to appreciate the importance of curiosity, observation, and perseverance in science. Today, we are especially honored to have Dr. Gyan Prakash Mishra sir as our speaker. Welcome, sir.
Thank you, Dr. Shiva. Am I audible, visible, both?
Yes, sir, you are audible, visible. Sir, I'll give a brief introduction to audience, sir.
Okay, you give. Otherwise, I decided to give myself.
Dr. Gyan Prakash Mishra sir was born in Varanasi and completed his graduation in agriculture from BHU Varanasi. He then pursued his post-graduation and PhD in genetics and plant breeding from Indian Agriculture Research Institute, New Delhi. He has been a Bioscott Fellow at both University of California and Purdue University, USA. Before joining ICR as a senior scientist, sir also served as scientist at DRDO. Currently, Dr. Gyan Prakash Mishra sir is a principal scientist and head of the Division of Seed Science and Technology at IARI, New Delhi. He has received many prestigious awards and also fellow of many scientific academies, including the prestigious NAS. His research focuses on marker-assisted breeding, particularly mapping of traits using advanced genomics tools in lentil and mung bean. He has mentored many students and has over 300 publications with nearly 4,000 citations with an H-index of 35. It's a true honor for us to have such an eminent scientist as Dr. Gyan Prakash Mishra sir as a speaker for our very first event. We couldn't have imagined a more meaningful and inspiring start. We are deeply grateful to you, sir, for joining us today to share insights on Mendel's foundation work and his continued influence on modern genetic research and discoveries. Now, I kindly invite Dr. Gyan Prakash Mishra sir to begin his presentation. Before we begin, I kindly request all participants to post their questions in chat box. At the end of the session, few selected questions will be shared with Gyan sir for discussion. Thank you, sir. Handing over to you.
Thank you, Dr. Shiva, and thanks all of you. I'm using some computer. I don't know. My camera is not working. I don't think it is required. Try 2-3 times, but it is going on and off. Doesn't matter if I'm audible. That's more important. Okay. And slide is visible. It's moving.
Yes, sir. It's visible and moving, sir. You
okay,
can proceed,
then perfectly fine. Okay. All discussions, everything will do fast. So first of all, thank you all. Thank you for this initiative. So what I decided, I was thinking that you asked that what topic to be taken. So let's try to understand Mendel's genetic legacy and why it is still important. So I chosen this topic of ever relevant and foundation, the kind of work many things, since most of you, or rather all of you, are from genetics background. I'm sure that you are all aware, because nowadays this Mendelian genetics starts from even 12th standards on. So let's see that what is very specific about Mendel and why something is very, very unique regarding Mendel, and in different ways. So if possible, we'll try to interact a little bit, because time is 1 hour. I don't know. It will be going passing. So let's first, let me finish, and then I'll be taking whatever question. So we all start our day with the data, isn't it? In the morning, we wake up and then we see what is the time, and then you are going to mess or wherever you are taking. So each day, we are playing with the data, whether it is mobile data or data time. Mendel also did this data, okay? And very different way and very, very unique way. Few things we know and few things we are going to learn or try to understand in this process. And how much this data is important, that we can understand that if you are taking data in terms of pooled value or if you are taking plant to plant basis, there where Mendel is little bit different, isn't it? And then using this particular data with the statistics, he was having some understanding about like statistics, and then he did with that data. Find this function. Fine. And then try to understand what Mendel has discovered and the recent context, genes and QT, in terms of Mendel. And then his Mendel and his principle, and his relevance in current time, 2020. Okay. I hope you must be having some understanding, at least like 7 chromosomes are there, and then all started when I was student, that 7 chromosomes, 7 traits on 7 chromosome. Then Dr. B.D. Singh sir book came, then 7 chromosome, these traits were sitting on 4 chromosome, then it now reached to 5 chromosome, and then finally to 6 chromosome, isn't it? So we'll be looking at that journey. And then the relevance. Everything is dependent on only two things, genotype and phenotype, isn't it? So, and then many times we get confused. Okay. See, classroom teaching is always better. I don't see in your eyes, I don't know what is happening, but still I assume that it, it is going the way I am thinking. So things are like genotype and phenotype. These two things were there. We are made up of whatever genotype is there, it is expressed. Sometimes your genotype is heterozygote or it could be homozygote. Okay. But if it is homozygous recessive, things are very, very clear. So sometimes we think that like if it is recessive, it might not be there in the population. So if you will be coming to Mendel afterwards, let's have a little bit understanding about these things. Fine. So if you see Western population, it is full of recessive eye color. Indian population full of dominant eye color. So it all varies. If we take totality, like complete population, it may be having P plus Q equals to 1. It may not be. We don't know. Okay. So with this, so many things are operating. And then Mendel did something way back in 1880, 65, 66. Before going into any detail, what we understand by genetics? We all understand, we remember this, like what is the definition of genetics? It is the science of heredity, which deals with the way traits are inherited from parents to offspring. This is what it is. So what we mean by parents? It's not like your immediate parents. It varies. It varies from like, it could be anything. To the generation, we can trace it out. It could be like 1 generation, 2 generation, 10 generation, whether it is human being or plant system, it depends. Okay. So that is the another reason we'll be saying that it is study of similarities and dissimilarity. How much we are similar or how much we are dissimilar among the individuals which are related by descent. And that now this descent is very big term. Okay. So if I have data for 10 generation, that is the descent. My like whatever way we are related, if I have the data, then only I can trace it out. And this is what this particular science is called. Okay. So 1865, this term was not there. Nothing is known, and things are moving in a different way. And then this science of genetics born after this Mendel. Not going into these terms, but still wanted to touch upon few terms. Sometimes I don't know what is the like size of audience and how much you are. So few terms I wanted to touch upon. For example, filial. Filial is a Latin word, came from filius, meaning son, and filia, daughter. So filial means irrespective of any like gender, it covers both filial, like son and daughter. So this is the way like F1, F2, F3. You can say F1, F2, F3 or whatever generation if you are knowing the parent. Okay. So this is how genetics goes upon. Okay? If you are not knowing the parental combination, taking any random thing, then we cannot use this particular kind of generation system. Okay, F1, F2, F3. If you wanted to be more precise, if you are maintaining that individual plants, then it becomes like F2 derived F3, F3 derived F4, isn't it? So this is the way we do understand this. Fine. Different terms are there. Mendel played with the characters which are only of two types, dominant and recessive, whatever he took. We'll be seeing the beauty of these 7 traits, that how different traits he took and how much he was lucky to have, like, even quantitative trait was behaving like dominant and recessive, especially for the plant type, isn't it? But then traits were not that simple. Traits are codominant, incomplete dominant, and then temperature-based expressions are there, and then even pH-based expression. If your soil pH is different, then also traits are different. Temperature, if you remember, there are cases. So traits are not that simple. Many times we say that these are non-Mendelian. It's not like that. We'll be understanding then how things were so integrated, like associated with each other. Broadly, what we understand, dominant and recessive, whether it is quantitative trait loci, then also this principle operates perfectly fine. Okay. So if you want to interact in between, we can do, but let's see. So Mendel was born, as Dr. Shiva was saying, actually he was born on July 20th. So you could have kept this on July 20th because that was again on holiday. So today, on July 22nd, he was baptized. Huh? With that child was like dipped in water for a few seconds, or a few drops of water was put on his head. And then this happens only in case of Christianity. Fine. And then there are few timelines. I find this very interesting, so I've kept it over here. Reference is also given. We'll be touching upon few major ones of this from 1882 till 1884, like once he was born till he died, and then we'll be reaching towards 2025. What is the Mendelian things which is going on even this year? Huh? One very classical paper, right? So if you see the contemporary, some scientists were there. Two names comes very prominent. One is Darwin, another one is, isn't it? So we'll be touching upon a little bit of this, that how much Mendel was smart and how these things were there. They were all contemporary. Still, it took time to understand what Mendel was saying, what he has published. Okay, and then rediscovery was. So, have you ever thought of anything which was discovered, published, and then he was struggling to tell that the world that I have already discovered these things, and still nobody understood? You see, he published his paper 1866. He presented his paper 1865 twice, February and March. Although these recipients were very less, around 40-50 persons were there once he was publishing, like presenting his paper. He still published his paper, and then he like took reprints, and then he has sent these reprints to 120 recipients across the world, whosoever he was, knowing that these are the person who might be able to understand. Okay, but then life was different at that point in time. So a monk was saying difficult to understand, but still unique, like lifestyle of Gregor Mendel. Right? So we write his name like Gregor Johann Mendel, but in Austria, they don't have j to be pronounced. They are saying like yeah. So he's not, the correct pronunciation is Gregor Johann Mendel. Yeah, no like j. Fine. So Gregor Johann Mendel, 1882 he was born, 1884, just like 61 years on this earth, and still he has made lots for this genetics. Actually, genetics was born because of Gregor Johann Mendel. Fine. And then in 1843, his name like Gregor was came because he joined monastery. So he took this name of Gregor in 1840. Fine. It was given to the monk like Gregor. He was not born with this name. And then he started teaching in a secondary school, but then it was forced to get like certificate for teaching in any school. There used to be some exam. So he failed first time and he again failed for the second time. So he was sent to University of Vienna for 2 years, 51 to 53, where he studied his physics under the professor Christian Doppler, who discovered this Doppler effect, and then chemistry, planetology, and plant physiology. Okay, so his mind has, it was like he has mentioned also somewhere, mind has changed during this particular period. He started understanding this crossing and other things, although he was working with his father since his childhood. So he was knowing crossing other things. And then once he went to this University of Vienna, because he failed in his exam, like he was passed, he was not sent to this. so that he'll be learning more and then he may get passed only for the secondary school. Then he returned back to Purno 1853, again continued as a teacher, secondary school. 56, again given the exam, same exam, again failed to become a qualified teacher. Fine. So he, like in during that point in time, if you read those literatures, so most of the monks, they are doing this. Okay, they are making crosses. He was not alone, like he is making. Other monks were also doing, because they have given this responsibility to like improve the plant species. Okay, whatever limited resources they used to have, because 24/7 they cannot be doing this. So they used to do teaching, a kind of like researcher, you can think. Okay, although not like trained ones. So he started his research using mice, but switched to plant because his bishop Anton was there. He believed that studying animal sexuality is inappropriate for anyone. So he was forced to like change his model from mice to the plant system. And don't think that Mendel only worked on garden pea. He worked on many crop species. But then, because of many reasons, which now we'll be looking at, and each and every book it is given that why pea was good. Don't think that like it's not like that Mendel worked only on pea. He was working with different other crops, especially these flower crops. But then managing pea, the kind of plant pea is a very small area. And since he has learned the statistics part, physics part, and then natural history part, so he preferred this garden pea. This is where the wisdom comes into the picture. That he has decided that I'll be doing all my experiments with pea, although he was working with other system, including mice also. Fine. So, and then next comes like 1854 to 1856. Although we find that whatever he has written in his paper, many things we are not knowing, because everything happened after his death. So we could not trace back any of his written literature. Only few pages were there that I have like included in this presentation. Right? So he started testing 34 P line, which he has purchased from the seed shop, whatever is available in his area, and then for tree consistency, 2 generations. So he has grown this for 54, 55, 56. Okay. And then thousands of plant he has tested, and out of these 34 line, ultimately selected only 22 line, what we now understand as homozygous line for the trait of interest. Because any individual like plant might be having 30, 25K gene, 25,000 genes. So whatever he was looking in those 22 line, he took, and rest line he has discarded because of some segregation which is going on. See that again, because homozygosity was not known, chromosomes were not known, nothing was known, but still he self it for 2 years and then took only those lines which were like homozygous in nature, which we know, right? And that point in time, he said that consistent trait was there so that he can use it for the crop. Okay. So 1865, he has presented twice, like in 2 consecutive months, February and March. And then he published his literature. Next year, although he submitted this paper 65 only, and ultimately it got published in 1866. Fine. And then this paper was in German language. Okay. And for the first time, it was translated to English, 1901. And after that, many like researchers, they have translated. Still, lots of like PhDs are going on, and then they wanted to understand what he has written, what was his mind, how Mendel reached to those conclusions. Okay, how much data he has used. So this is what is this paper, easily available. So English versions are also available. If find time as a genetic student, at least we must read English version and then see what kind of person he was. And then he took more than 10 years to do the research without anybody's help, without any funding, without JRF, SRF, without student, and then reaching to this level of writing paper, presenting his work, sending it to more than hundreds of person reprints, and then like ultimately he said that one day we all are going to understand what I am saying. Right? So why Mendel was unique? For the first time, somebody has applied statistics in the biological research. Never ever done, and that was the problem also, because nobody is able to digest that what he is saying and why he is applying this kind of. One thing we always miss, always try to keep in mind that data, what the way he has taken was plant to plant basis. Generally, what we used to do, we'll be pooling the data like F2, F3, but he has kept individual plants data differently. And then only it is possible for him to discover all those what we now know as Mendel's inheritance. Okay? So, and one very, very unique thing is that Mendel discovered laws of genetics without any hypothesis. Okay? There was no hypothesis. Nobody was knowing. He was not knowing. And he derived whatever he, like data he has taken, based on that, then hypothesis was formed that this could be, okay, particulate kind of inheritance. So his laws were discovered without any hypothesis. Nothing was known. It's the F2 data which has resulted in the like what are those laws of inheritance, which we have proposed. He has never written. He has written a paper from where we have like derived these laws. Fine. And the biggest problem was that since he was a monk, so he was always considered outsider by the scientific community. But that time, scientific communities was very narrow, isn't it? So it was not no WhatsApp, no online, nothing. So if you are knowing one language, it is very difficult to even to translate into another language. You think he has written his paper 1865, 66, and then it got translated for the first time 1901. So that much of the scientific gap was there. And then since being a monk, nobody considered like he was much, much ahead of his time. And then see, but Mendel was knowing everybody. He was knowing that Nageli is working, and then he corresponded again and again with the Nageli, Carl Wilhelm von Nageli. He was there in Germany. He corresponded in 1866 to 1873 many times, and he wrote letter. He has sent his manuscript to him, and he was competent enough to understand the significance of his work. But again, Nageli was very much preoccupied with his own understanding, so they were not ready to accept what he was saying. Although Nageli said, okay, let's do one more experiment with you, and then that has created problem. And then ultimately, he died very early, like just only 61 years of age, and again Bernoulli. Okay, so Mendel was not only like monk; he was a good reader. He was reading different books, and one book which has inspired Mendel that was from Carl Friedrich Gartner's book, Experiments and Observations on Hybridization in Plant Kingdom. This book was published in 1849. Mendel was having copy of this particular book. And then if you see this book, mainly it describes more than 10,000 experiments on plant crosses, mainly interspecific crosses. So Mendel, what we know that he worked on 7 trait, one as it's not like that. He also took many interspecific crosses. So he also wanted to, like Mendel's main aim, if you go into detail, then you'll finding that he was crossing different species of Pisum, and he wanted to have a new species to be derived. This is what that point in time concept was going on. Okay, he started with this logic, and then he found different types of trait, different types of data, and then altogether different kind of genetics comes into it. So this is what how Mendel has written in that book. I'll be coming to next slide. So Mendel was having this book, and it is full of annotation. Whenever he reads book, there are few blank pages, so he used to write something. Okay, and then end pages he has made 12-line note. This is first 12-line notes was there of characteristics of 5 Pisum species. Different species were there, and which he used in his paper also. 1866 paper, he named those species. You see, we'll be coming to next. You see, he has written Pisum arvens, then Pisum sativum. So he has mentioned 5 different species: arvens, sativum, umbellatum, saccharatum, and quadratum. Okay? And then right hand page, because few blank pages were in that book. Okay? So then he has described the characteristics of hybrid between these are 2 flowering plants, Jam and Equilegia. And then he has made cross of this also after 1863. Okay? So Mendel could have made this note. It is proposed that this notes, whatever is written in this last page, while he was preparing for his 1865 lecture. Okay, since it was a book, it is preserved. Whatever Mendel's notes were there, since he died so early, and then he that monastery they have cleaned everything, so nobody could retrieve Mendel's original notebook. If that was like found, we always think that he could have like discovered linkage also, but it was not known. And if you see Mendel, so it is not like that Mendel was unaware and he's just doing this. Mendel was knowing everything. Mendel was well like aware of what he is finding. It is the problem of others who could not understand. So this is one like a newspaper which was published way back in 1961. In that, if you see, so it is written that July 26, 1861. It was written that Father Gregor Mendel, professor. This was the translation of English translation of this news cutting. That he was aimed to of improving vegetable and flower varieties. He was working on many things, and then a credit was given that he has he got some one which is very good. Then he tried to popularize that also, that by grown by professors such as fish oil, cucumber, bean, many things he was working. Okay, and then he has temporarily extended his experiments to several species of flower. But then ultimately, once he started understanding genetics, kind of a thing in pea, then he changed his focus only to the pea. Okay? But he was publishing. He was calling different newspaper, and then he used to publish what I am doing, what is good for the society. Okay, and then he also, it is written that buying seeds abroad can better preserve for domestic production. So the kind of varieties he'll be developing, that is going to save exchequer also for the country. Okay? This is 1861. Now, if you see during that period, what is the problem with Mendel? Huh? So if you see, not contemporary in the true sense, during around 200, 300 years, 1590, around 1600 to 1880, okay? And we'll be always keeping in mind 1822 to 1884, when he was alive. Huh? So 1590, if you see some major ones, huh? So microscope was developed for the first time. This is how this microscope looks like. That was the situation. Forget about your mitosis, meiosis, or anything. Okay, and that too not like in this generation of internet that you discover today, and today only I'll be knowing. So things were not that. Then 1667, Robert Hooke published Micrographia, details of various using this microscope. Then Leeuwenhoek first time observed the bacteria, 1675. Then Nageli discovered subcellular structure, later known as chromosome. He was only saying that I find something. Even in 1888, Valdayer, he has coined the term like chromosome. So he was not knowing what this chromosome. Chroma, that is the made up of two words, chroma and soma. So chroma means like thread-like, soma, chroma means color, and soma means thread-like structure. So he find something which is thread-like structure in the nucleus, so it is named as chromosoma. So till 1888, these things were not clear. Okay, everything changed around 1900. By that time, better microscopes were there, chromosomes are known, mitosis, meiosis were known. Then it took like very easy for 3% to say that this is what I have discovered. So if you see, it's very small plot, 115 to 23 feet plot is still at that part of they used to maintain. And then if you go and see, it's still this particular, it is said that it is the same plot. Okay, so it is being maintained during this period. Total amount of data it was calculated, it is touching somewhere like 30,000 plant data. Okay, no PhD, nothing, no masters, no guide. So he has taken that much meticulous data, and then based on that, then he wrote a paper. Again, the next very important part is what? Your cell theory. Cell theory, which came in 1838. Again, we are not reached to then 1865. Okay, it says that cells are universal component of organism. And this cell theory of Schleiden and Schwann, it says that cells emerge de novo. Okay? By 1850, it came to this concept that no, it is not de novo. They are coming from pre-existing cells. Till 1854, even fertilization was not clear. Okay, so at that point in time, what was the mechanism? It is believed that embryos develop from egg cell. After just there is a contact between egg and pollen. No fertilization concept is there, double fertilization, triple fusion, nothing was known. Okay, so in this backdrop, Monk is working and then proposing that something is coming after generation, how these traits are reappearing. So it was unacceptable to the scientific community. Okay? So now we reached from 1590, 1888, things were not that clear, but things are getting clear day by day, isn't it? So what was Mendel was forced to say that no traits are inherited as particle, because this is what he could understand. And like he was very firm believer of his own data. Okay, we'll see why he was that much clear about his data. Then he said that my concept is particulate hypothesis, and parents are passing some discrete heritable unit, now we know as genes, to their offspring. Okay, and this by this only we can explain the reappearance of trait. Here means your recessive trait after several generations. If parents are homozygous dominant, homozygous recessive, so reappearance can be explained if there is some particle, isn't it? So did not know that in due course of time, it becomes chromosome, DNA, or ultimately the gene. Another bigger problem is that the contemporary, what is the big scientist? At that point in time, Charles Robert Darwin was there. You see, he's 1809 to 1882. Mendel was 1822 to 1884, isn't it? They are completely contemporary. Again, he was from UK. He is English person. So whatever he is publishing, and that he was dominating at that point in time. So he proposed pangenesis theory that each organ and tissue contributes tiny parts that are like collected in the sex organs and determine the configuration of it. And then once he has said, so it is final. Let's say touch upon few things because you all know, so it is not like much to be like discussed, but still why Pisum sativum? As I said, Mendel was not working on only Pisum sativum. He was working on many things. But the reason could have been what I could understand, the easiness of like his trait. You see, like pod color, pod shape, then seed color. So all these things can be harvested easily and kept. And then whenever he finds time, he can take the data, isn't it? Out of his like 7 trait, only data which is required to be taken into the field condition is your flower position and then plant height. Rest all can be taken easily any point in time. And then very easy to grow from one plant. He'll be harvesting hundreds of seeds, which is basically required for any statistical analysis. Then comes your growing quickly, small area, lots of offspring per plant. Okay. Then purity of plant can be done. It is by nature like self-pollinated. And then kind of trait what he has taken and artificially cross-pollinated. If you start crossing lentil, very difficult. If you wanted to cross pea, very easy because flower size is very, very big, isn't it? So if you see seed color, ripe seed form, mature pod color, ripe pod shape, stem length, flower position, flower color. So flower position, stem length, only these things like it is needed to be taken. Otherwise, rest all can be taken once you are free. So you can just have harvest it, pull it, and then you can do it. So he started with 34, then 22, then finally studied 7 traits, which is published. Okay. One trait you can always find some book, something is written that is flower color and seed coat color. Okay. So flower color, purple and white, and seed coat color, gray and white. Okay. These two are like pleiotropic trait, not linked trait. If something is linked, it can be broken. Linkage always can be broken by crossing over. You increase the population size, it is broken. But here it cannot be broken because same gene is regulating two different ways. Huh? So always remember that these two are pleiotropic in nature, controlled by single gene. Okay, and then he counted offspring of each phenotype, analyzed the results mathematically individually. He never mixed any cross data. Okay, if you have time, enjoy these like numbers. So I kept this for a certain reason. You see, smooth seed and wrinkle seed. Number of plants, number of seeds he has taken, around 5,400 smooth, and then this much as a wrinkled. Then yellow seed, green seed in thousands, isn't it? But if you see green pod, around 428, 152, that comes to around like 550, 70, 80. So one pod, if it is like this much of seed, you can calculate by his data that how much he has taken. But he has made sure that whatever number he is taking, that is statistically significant to do any kind of analysis. Okay, that's the beauty of his data. But unique part is that you see how much close it is, 2.961, 3.01, 2.951, very, very close to 3:1, whatever data he has presented. Okay. So if you find time, do the chi-square analysis and see how close it is after looking at chi-square. This paper came in like 2001, okay, by Reed and Ross. So wherein it is mentioned that these are the 7 traits, then the symbol group, then the linkage group will be coming to chromosome since chromosomes are now known clearly. Okay. And then what are the gene function? So till like this year, only 4 genes details were known, means they are cloned. And this year, now one paper came in Nature, which has clearly identified all the 7 traits and exact location on this chromosome. So we'll be looking at this paper in the last. So till last time, I used to tell that only this many are cloned. All the gene functions were known, and still those genes are seen. But now this cloning part is over. Now we are knowing precisely this is the gene. Okay? Meristem function, sclerenchyma formation in pod, chloroplast structure in pod, then BLHS transcription factors, stay green gene, gibberellic acid pre-oxidase, starch branching enzyme. All these genes were there, which is regulating. And it is such an irony that genetics was born because of this particular crop, and still we have struggled till 2025 to find that exact location, function, and the cloning of this Mendelian 7 genes, isn't it? And if you see the size, hardly 3.2 GB is the genome size. It is not that big if you compare wheat and other crops. Okay, so but fortunately we do have. So don't not going to touch, but still wanted to touch upon few. If some students are there, law of dominance credit was not given to Mendel because it is already known at that point. Credit is given only for these two, like segregation and independent assortment, because that was not known. Okay, so never ever like we consider this as a Mendelian law. Fine. F1 offspring of monohybrid cross of a true breeding parent resemble only one of the parent. This is what it is. If you make a cross, one is coming. But in due course of time, we'll be learning that things are not that simple as they used to be. It's not that simple that all the traits are not behaving in this way. But still, I feel that they are following Mendelian genetics. Okay? Even your QTLs are following Mendelian. Little bit about law of segregation. Factor remains in pair. Now we know that genes remains in pair in the hybrid or F1, and during gametogenesis, they separate or segregate. Very, very important thing is that without contaminating each other. Okay, to the gamete. If they contaminate, it becomes exception. What are two exceptions mainly? Non-disjunction and meiotic drive. Non-disjunction, if genes are going together in one, another one is like free. And meiotic drive means your dominant becomes recessive, recessive becomes dominant. So your total number of like gametes, they are not equal in number. Okay, so it drives your meiotic drive is there. And I don't want to go in detail. Hopefully, if you know, if you don't know, I'll come some other day to explain more about this. Okay, so this is what is called as law of purity of gamete. They won't contaminate each other if two allele of a genes are there. If it is diploid in nature, if it is like polyploid, things becomes different. Fine. Independent assortment. And one more thing, like from Mendel's paper, these laws are derived. He never written like this is my first law, this is my second law. Even he has not written that these are the laws. Okay, but based on his paper, we came to the conclusion that these are the things which is mentioned, and now it is considered as laws given by Mendel. Right? Segregation of one factor pair. Earlier we are dealing about only two genes, the allele of a gene. Now here it is one factor pair. Could be like multiple factor pairs also, dependent of segregation of other factor pair. If it is not independent, it becomes linkage or sex-linked gene, isn't it? So if something is linked, so it becomes like exception to that. So wherever, so it is just exception. This does not mean that they are not following Mendel's law. He has never said that this is my law. If that is the situation, what 7 trait he has taken, it is going to follow same rule. If 6 linked genes are there, another component is not there, then it is not behaving. So again, that is like confirming whatever he has said, isn't it? So if we come to the reasons of success, a few very, very common things were there. Why it is successful? Selected characters in two distinct forms. Although 34 lines which he have selected, out of which 22 lines which he finally decided to work, they are having multiple forms of a gene. But he decided to select only two. For example, flower color. He decided I'll be crossing only two. For plant height, he has decided different heights were there, but he decided I'll be taking only these two crosses. He might could have like taken many other crosses, we don't know. But whatever is mentioned in his paper was only two. Okay, and then he used true breeding P genotype, and then he took 2 years to do selfing and then confirming that these are the things. Okay, all those things were never ever mentioned. We don't have those like literature in which he has written by his hand. Otherwise, like could be have been a treasure for like geneticists times to come. And then he used experimental approach. Each year data he is maintaining. Okay, like first year I made the cross. This is F1. This is F2, F3. He went for multiple generation, and then only he did statistics to study the natural phenomenon. And then his like experimental planning was very, very meticulous. It is said that when he went for your studying, at that point in time, he was studying different book, and then only he has decided that I'll be doing something. It's not like that one day he has came with this thinking that I'm going to do, I'll start planting. It is not like that. Okay? He has critically recorded the traits for different generations. Many generations he has recorded, and then he analyzed it. No gene interaction epistasis. Again, he was lucky. No linkage. We don't know whether he has found linkage or not. Whatever he has reported, no linkage. And then his childhood experience was there of grafting of tea. Like his father was doing all these things. He was always there with his father and trying to learn. He has learned very, very like carefully how to do this crossing, male and female understanding he was having about the plants. Okay. And then his university experience in physics and natural history. That has changed everything. Okay. And then he was a very firm believer that world has an orderly place governed by natural laws. And this concept has forced him, or he has given the liberty to start using mathematics in case of biology, okay, which no one was thinking and forget about doing. So during this particular period, 1854, 1863, around 9 years, he cultivated and tested 30,000, more than 30,000 pea plant and maintained the data, then analyzed the data. Then let's see this Nageli first part. So 66, once he published his paper, he sent that reprint to Nageli. He was at that time like professor in Munich of plant physiology, along with a very big letter, okay, mentioning that he intended to repeat his experiment with other species. And when Nageli was understanding about genetics, he was having some idea. So he said, okay, let's take Hyraceum. Hyraceum looks like this. So he said that let's do it. Again in 1867, Mendel wrote second letter mentioned to Nageli, and wherein he mentioned that it was not easy to reconcile with the contemporary scientific knowledge because by 1 year he has already sent 120 letters, and he was the kind of like response he was receiving. He find that I'm receiving mixed reaction to my lectures and to my research, and nobody is accepting, including even Nageli. Nageli only said, okay, let's repeat it, but he was again not much interested in like doing this. And then Mendel has written once that called himself in his letter that I'm an empirical worker. Empirical means from the data I am deriving something. Okay, I am not going with any concept. From data I am deriving the concept, and described his experiment as empirical. Empirical whenever we do, like we do something, getting the data and then going to some conclusion. So although Nageli understood that he studied about inheritance, but since he himself was firm believer of building inheritance, and then there is no point in time, like he cannot understand what he's saying, but still he like worked with him on Hyraceum, and you know that because of this apomixis, not all the plants, that ratio was not fitting, and then Mendel also decided to leave this. Same time, another person was your Charles Darwin. So he was a well-established scientist because he is using English. He was from there, and then he like on the origin of species he has written 1859. Okay, an observation for 5 year voyage. We know that he used this HMS Beagle, and then he moved across the world, and then he got some macro concept of evolution, natural selection. Then he wrote a book, a big scientist, and then he proposed ill-defined like claim. Ill-defined means he said that like begets like. So if you could be looking like your parents, huh? And something comes, and then things were not that clear, and struggle to understand not what we now know as transmission. Okay? And then he proposed a theory, pangenesis, 1868. Mendel publishes like his paper 1866, 2 years before his like theory. And Darwin never encountered Mendel's research. Mendel has sent his paper to Darwin also. He sent it by post. And then once like Darwin was no more, then it was found unopened in Darwin's shelf. It was lying there, but Mendel, like Darwin, never read Mendel's paper. So same time, two theories are coming, and there is some communication gap because of language, because of many other reasons. And then again, Mendel got promoted to Abbot. Abbot means a little bit higher position, 1868. So 65, he presented 66 after 2 years, and then he was engaged more of doing like monks' work, like monasteries' work, rather than doing science. Although he was doing science, But since he was not able to find with this Hiratium experiment, so he got a little bit disappointed. Nobody's understanding. He is trying to send, and he did whatever best is possible, and then he left. Like typical doing science, he did not publish much. So natural selection, Mendelian genetics. Luckily, you see, both are there at the same timing point, but never intervened during their lifetime. So Mendel was knowing who is Darwin, but Darwin never, he was not knowing who is Mendel, although Mendel sent his paper also to Darwin. Okay. And these two is now the reason for foundation of evolution. Some good papers are there. I have all mentioned over there. Okay. Then comes your 1900. Okay. When this rediscovery happened. So Mendel read first time 8 February, then March, I do not get the date, that which date he read. And then hardly audience was 40. And 1900, 16 years later, okay, 3% independently. Dutch biologist Hugo de Vries, German plant geneticist Karl Quarens, then Austrian plant breeder is Sharma. They independently found something and they wanted to take the credit that we are the discoverer. But by the time they found that no, Mendel has already done, and then all these three were given this that you are now rediscoverer. You see, whenever Mendel comes, then comes the rediscovery, and then these 3% they are also always get the credit of rediscovery. Have you found any such thing which is like rediscovered? If you do anything, it is discovered. Okay, but Mendel was much ahead of its time, so that it took even 16 years later of his death, then only rediscovery came into picture. Otherwise, it took around 34 years to understand what Mendel has said with full confidence. Mendel was fully knowing what he was saying. And then once this rediscovery came into the picture, then we could find only a few letters written after 1866 to Nageli, what Mendel has communicated, and which is professor in botany. And then Nageli's response were published by this Karl Korens. He was knowing that what are all those things were there in 1905. Otherwise, nothing was there. As I said, after the 16 year of his death, no literature, nothing. He was not having any family. He was just a monk, living in one room. So everything was discarded. Had it been 1 or 2 years, or had it been he was living for more than 70 years, things could have been much different. Okay. So Bateson used fowl to demonstrate Mendelian principle. Then all sorts of model organisms were used after 19. And then different terms were coined. So Bateson was very fond of coining the term. So for the first time, he coined the term genetics, which in Greek means geno, to give birth, a personal letter to Alan Segby. And then he used publicly Third International Conference on Plant Hybridization, London, 19. After that, this term was finalized. And he also coined zygote, F1, F2, allomark, allele, epistasis. Still, we are using all these terms coined way back 1905, 1906. Okay. He worked with Punnett, and then William, again, here nothing if we pronounce it will be Johansson, not Johansson. Okay. So Wilhelm Johansson, he coined the term gene, genotype, phenotype. India, you can always pronounce Johansson, otherwise they'll say that what is this? So if you see the target audience, accordingly you can pronounce, otherwise it is Johansson. 1889, de Vries, he coined the term pangene for the same concept. So different. The moment 1900 came, science of genetics changed like anything, and it is still changing till 2025. Why ignored? Many reasons. Some basic reason what we consider, published in obscure journal, lack of understanding about the chromosome, Darwin other looking for continuous variation, probability mathematics, unfamiliar. He did not publish much. It's okay. So many things we say. But if you ask me, biggest thing is he was much ahead of his time. Okay. And even if he's saying he is printing, he's sending his transcript to others, still nobody is ready to accept. You can think of how much ahead of his time Mendel was. Okay? That was the reason that nobody could understood. But basic reason we can say that these are the major reasons. If you see 19, I may take some more time, okay, may not be able, so it's okay. Or
Yes, sir.
you
Yes,
want
sir. You
me
can.
to finish? Okay, not much, but
Take.
I'll be just going. So Carl Fisher, 1936, again a Britisher, he reanalyzed the Mendel's data using 10 F2s and found that that the kind of ratio Mendel has proposed in his paper, he said that it is too good to believe. Okay, another story. So we are not going into that details, but still you'll be finding something. Why? What is the application? If you ask me one line, predict the outcome of process between different strains of organism. Okay, different methods for their Punnett square, Hope, probability. So what you are predicting based on Mendel, you can predict if you know the number of gene pairs, you can predict kind of gamete, kind of population size, genotype, homozygous F2, like heterozygous F2, type of F2 phenotype, all different like formulas were there, which is very easy to understand. So based on Mendelian genetics, this came into the picture. We can predict what is going to happen. If you know one gene, two gene, three gene, QTL, major QTL, minor QTL, if we know, we can predict. Okay, we can predict the population size, how much we want. Then another good paper, or you can say that unique paper came 219, when for the first time reference genome of P was published. Okay, and then it is the first annotated chromosome level reference genome was there. Hardly 3 GB genome size, but still, you see, 219 for the first time it was sequenced and published. Okay, and then it showed the genomic arrangement across the legume, and then it has also suggested major role of repetitive element in P genome. Whatever genome of your it's not only P, lentil, wherever genome size is touching around 3 to 4 GB, you'll be finding a lot of repetitive elements are there. Okay, no one knows why it is there. In times to come, we can surely going to understand. So this paper confirmed the previous interpretation for the four characterized Mendelian gene. Okay, R, L, like I, L, E, and A. Okay, still that three genes are left, which get due recognition in 2025. We'll see that paper, very, very classical paper. Then another paper came 7th November 2022. It published in Molecular Plant by Shefeng Cheng. Again, chromosomal level clarity came into the picture. That chromosome 1 is earlier, which used to be called as linkage group 6, then chromosome 2, LG1, chromosome 3, LG5, and then different gene name came into like P, I, R, GP, FA. Okay, different names were there. Then this year, 2025, paper came. It was termed as Genomic and Genetic Insight into Mendel's P genes. Okay. So what they did, this Feng and co-workers, they took 697 P variants, and they have generated 60 TB data. We generally say that if genome size is very big, how to do like GBS GWAS? Very, very difficult to do. But they have generated huge amount of data, and till now, whatever I find, this is one of the best paper to be referred in the class and to understand that even GBS GWAS can be done using such a large number of genotypes, 697P variant. They have generated 60 TB of data set, and then their focus was mainly those three uncharacterized states. What are those? Pod color, pod shape, and flower position. They were still uncharacterized. I never thought of that they are going to be characterized using this mechanism of NGS. So pod color, they found like 100 kb deletion is there upstream of CH chlorophyll G synthase gene. Then pod shape, it was found that it is because of the mutation in MYB gene, which is causing parchment-less pod, which is giving you shape, like constricted or plated. Then flower position is because of fasciated stem phenotype. It has nothing to do with your flowering. It is the stem phenotype which is changing the flower position, that whether it is terminal or axial. Fine. And then not only this, they have recorded phenotypic variation for other traits also. Let's see. You might have seen like so many Manhattan plot, but this unique Manhattan plot of this first we see the trait, round wrinkle. Okay. And then you see the log 10 p value. They have used 697 variant, 60 TB data set. It is touching somewhere like 200. Okay. We sometimes struggle with few numbers. It went up to 200. And again for stay green trait, which is giving your cotyledon color, whether it is green or yellow, it is going somewhere 80. Right? You see 60. So that is the like confidence level came with this particular analysis. Unique case, it published on 23rd April 2025, and then exact details were found that how much insertion, how much base pair, where it is, just because they have taken large amount of genotype and very huge amount of data is being generated. You see, There is no confusion at all. Just one peak going very, very sharp peak. And then sometimes we don't get such a good peak even with our RILs. The kind of peak was found with this. Okay. So what it shows that any technology is good. Only thing is that how much amount of data, how much precision in your phenotyping is there. That is going to give you such kind of data even using your GWAS with this kind of method. Okay. Again, for your stay green trait, T1Y copia insertion was found. Again, then for flower color. Okay. So all these details were identified, which is earlier not that clear using with this. Then we are already knowing up to this. So three gene color of unripe pod, shape of ripe pod, and position of flower. Again, it was found. You see, just a peak. LE peak is there. LE generally it is said that for length, this LE was used. Otherwise, it is not clear. These terms were not very, very clear. GP is for green pod. There are two genes uniquely, if you remember, for your constricted and inflated pod, there are two genes. One you see P over here, chromosome 1. This at the bottom, these are the chromosome. And then on chromosome 5, B is there. Okay, so P is for pod shape, B is for pod form, both are same. So either of that, if it is present, it is going to give like what is will be your right pod shape. Okay, so out of this large data set, they have identified these two genes also for same one single trait. Color of unripe pod, again, GPG very, very clearly. You see the log 10 p value 300, 50, 60, 300. Okay, unbelievable. So such a good data they have generated. Position of flower, fasciation is there. So fasciation is a stem like of a trait, which gives fasciation to the stem, and it determines the position of flower. Okay, so all this confirmation was done using the latest 2025 paper, which is going to be referred for times to come. Little bit long, let's shift to clear this confusion about why then QTLs, whether QTL is part Mendelian genetics can be like cleared with the QTL. Not going into much detail, we'll be touching little bit about this. There are two things to understand QTL. DNA, chromosome, gene, QTL, we understand. Dependent variable, take one example. We can take any example for disease resistant anything. So yield per se has no meaning. It is dependent on many other things, isn't it? Chlorophyll content, how much, plant height, number of flowers per plant, number of flowers per peduncle, seed weight, all many other traits are there which will be giving your yield. So if you want to map yield, you have to independently go to these traits. Now we need to understand how these traits are varying. And then my trait comes into the picture, which is called dependent variable. Independent variable, dependent variable. Okay. So let's take this example. So earlier, like AA, one gene, suppose it is regulating one trait, not two trait, three trait, okay, just one trait. So three different groups. If two genes again regulating one trait, let's take example of skin color or height of an individual, plant height, human height, could be anything. So again, different groups are there, 9331, okay, although my trait is same. Again, if there is three loci, 27999. If you go on increasing loci, again, we can think upon like one is controlling chlorophyll, another one is controlling something, another one controlling partitioning, and ultimately it is resulting in yield. So we'll be having different combinations. This is how QTL and gene comes into the picture, isn't it? If it is more than two, it becomes QTL, quantitative trait loci, which is regulating my trait of interest. So again, QTLs are nothing. It is chromosomal region only, which contains alleles. Sometimes number of genes are very high. Okay, sometimes only a few genes are there. But we need to first reach to the region, and then we'll be going towards the identification of my gene of interest. Okay, fine. So again, how to identify QTL? There are two strategies. One is QTL mapping using mapping population. It could be biparental or multiparental mapping population, or it could be LD-based mapping. LD is called linkage disequilibrium, using Genplan panel, association mapping panel. Again, whether it is your mapping population, Genplan, in each case, it has to go through the crossing program. It has to, whether it is natural crossing or you are forcefully crossing, the way Mendel has done. Only thing is that Mendel's trait were very, very distinct. Here, if you are mapping a loci which is contributing to trait of interest. Mendelian principles are operating there also. Only thing is that how much PV is associated with this particular trait. Otherwise, when if you are using association mapping, there also it's like historical recombination. If there is no recombination, you cannot. If there is no recombination, Mendel could have not found anything, isn't it? So what we require? Mapping population, contrasting parent crossing, same Mendel did, saturated linkage map, then reliable phenotypic screening. If your phenotypic screening is not reliable, you're not going to get anything. As Mendel has found, even tall and dwarf, he was very clearly distinguished that this is like how it is segregating into 3:1. Okay. And then what is the process? Contrasting homozygous line. Same Mendel took. Then trait phenotyping, replicated trial over location, and here what he did, parental polymorphism, if we are going with the marker, genotyping of individual, framework linkage map, and then you are going to find association. So we saw like 2011, we are struggling. We are knowing only 4 clone gene. By 2025, things were much better known than what was published like in the genetics paper. Okay. So multiple regression is a term. If we try to understand QTL. So a statistical technique which analyzes the relationship between a single dependent variable. Sometimes we struggle with this terms. Dependent variable, always keep in mind, I'm talking about yield, disease resistance, and several independent variable. Markers, QTLs. So chlorophyll content, it has to be linked with marker. Then only it becomes QTL. Plant height, number of flowers, seed weight. It could be anything. Partitioning of photosynthesis. So all this is ultimately going to determine. Out of this, whatever is major, it becomes major QTL. Whatever is contributing less, it becomes minor QTL. And we measure in terms of PVE. Mendel measured just like qualitative measurement. Red, pink, white, purple, whatever. Tall, dwarf. Here things are not like that. Although we are measuring here also, but we are measuring in terms of phenotypic variance extent. And then different ways were there: single marker, interval mapping, composite interval mapping, multiple interval mapping, then QTL-seq, BSA-seq. Whatever you are doing, you will be knowing your two parents, making crosses, generating population, and then reaching to your rate of interest using markers. If you see Mendel's discoveries and modern plant breeding, progress in plant breeding is essential to understand the traits in inheritance. Okay? Without discoveries, Mendel discovery, earlier also everybody was doing arbitrary crosses, selecting the most appealing plant. But this phenotype-based approach and then use of molecular tool would have not been possible if things were not that clear. Okay, and then now we can even predict the trait, isn't it? We can predict the trait. We are going with the phenotypic selection using molecular tool. We can combine the trait. Okay, so predictive genomic models were there. But whatever we do, we'll be still dependent. If you're not making crosses, we cannot do anything. Any breeding strategy, whether it is modern or even times to come, whatever way you are selecting, that is another way that I can select based on computer that I'll be taking these two parents because I'm having complete genomic data with me of 1000 line, 2000 line. But then logic is going to be always same. It has to be contrasting. Okay? Once it is contrasting, then we will be doing some good genetics as Mendel did way back 1860. You see, the data is now changing. Big data. When I was student, then Human Genome Project took 13 like years. Billions of pounds were used. Now you can sequence your whole genome maybe in hours. Okay. Then deep learning came, machine learning came, and it's all play of data. My MSc, I remember around 30, 35 SSR primer. 6 months I was struggling. Now we are playing with like GBs of data. 50 GB, 100 GB, even for the masters. Okay, so things are changing. We need to change with that. But basic genetics, basic of Mendelian genetics will going to be the remain same. One has to understand whether my data is deviating, what is the reason, why it is not following Mendelian rule. We need to find the reason. Okay, if there is large amount of input data, genomic, proteomic, metabolomic, clinical data, simple models may fail. So we have to go through deep learning kind of a data. And then for any good data generation, your parental combination, making crosses, mapping population, phenotyping, genotyping, and then how good you are in that, then only things come. This machine data is the last. First is the selection of parent, generation of good population, and then only comes the sequencing and then use of this. If you are losing at the selection of parent, nothing is going to come in your hand. If you see, these three are very, very important in case of like evolutionary biology. Now things are not moving in isolation. So theory of heredity, Mendel, 65, 66. So just two different combinations were done. Then Charles Darwin, he was studying the things at the level, he was understanding or proposing things at the level of species, isn't it? And then your Charles Elton was there, he's studying the things at the level of population. And all these three started merging by 1930, and then new synthesis came into picture, and then a science of evolutionary biology was born. And in due course of time, whether it is Mendelian theory or origin of species or population ecology, different scientists constantly contributing, and this is still evolving. It's not like that we have understood everything. Still, we are struggling to understand rice genome, which is very, very small. Okay. So to conclude, if you see, Mendel's experiments have laid the foundation of modern quantitative genetics. Okay. We are part of that. And a few months before his death, and he was died just because of your chronic nephritis, no? That is, and it has, which has resulted in heart and kidney failure. That point in time, things were not that much. Medical situation was also not there. He was in, and like, and what he like told during his investiture of his successor to Franz Barnia as abbot, he reportedly said that my scientific work has brought me great joy and satisfaction. He was completely satisfied. Only we have not understood, but he was knowing that what I have done is remarkable. And I am convinced that it won't take long that the entire world will appreciate the results and meaning of my work. So few months before his death, even in 1884, to his successor, he told like this. Okay, and unfortunately, the impact of like his whatever he was trying to convince to the world, it took like 34 years after his discovery that whole world started understanding, and he has given this like his father of genetics. If you see the impact of his discoveries for agriculture, evolutionary biology, precision medicine, his is not like one or two area. Even 150 years of his publication and more than 200 years of his work is beyond any quantitative measurement. There is no measurement available. Whatever we do, whether it is quantitative, qualitative genetics as like nothing. Whatever we see, there is a pathway. No? Even Mendel has found tall and dwarf. We know that this particular gene is there, but everything is pathway. Okay, so if we are studying at the last gene, if that mutation is there, it becomes, you may find that it is qualitative or quantitative. If you start measuring like flower plant height in centimeter, becomes quantitative. If you are measuring tall and dwarf, it becomes qualitative. Okay, so of course, major QTLs were there, major loci were there, which clearly distinguish a population as qualitative or quantitative. Okay, so this is how like Mendelian genetics is growing. It is still growing, and in times to come, we are going to learn much more on different science like sphere, whether it is like human beings or any other area. Okay, so with this, I completed in 1 hour 10 minutes. Thank you all for your patience, hearing, and whatever you wanted to ask, now you can ask.
Thank you, sir, for delivering such an inspiring lecture on Mendel and also brief history on genetics. You truly helped us to connect Mendel's and his groundbreaking work, and also helped us to understand how these genes are mapped and understood. And your depth of knowledge and engaging delivery made this lecture a profound tribute to Mendel, sir. Thank you, sir.
Thank you.
Participants can
Thank you.
unmute and ask question.
You all are scientists or like students also here?
Sir,
Mhm.
sir, the group consists of scientists, students,
Okay.
uh, breeders who are working in private firm, but most of
So,
them are students, sir.
Bhaskar, you are asked, na, do this linkage group change in future? See, now concept of linkage group has gone. Now things are knowing, we know that now chromosomes. Okay, so no question of linkage group. Now we are exactly knowing that these traits are there on 6 chromosome, chromosome number 7, there is no trait which Mendel has studied. Okay, and forget about this 7 trait. Now this paper, I must request, this is like open access paper. Download, you can see they have now mapped hundreds of traits. Okay, such a good paper rarely comes. So this is not going to change. This is now final. Genes are final, your mutations are final, and then chromosomes are final. Fine. So no issue at all. Few things I skipped because I'm told that it is 1 hour, so I don't want to cross. Okay. Sometimes what happens if I'm only speaking because I want to see in eyes, the classroom is always better. So this linkage group is not going to change. Okay. And now we'll be using not linkage group, but see, don't get confused with cross-pointed crop. If we want to study genetics, okay, whether it is cross-pointed, self-pollinated, it has to be homozygous in nature. For what? For the trait of interest for which you are studying. You cannot study 25,000 traits at a time. We cannot think that even Mendel has self, so it is 100% homozygous. Nothing is 100% homozygous. But the trait of interest which I am going to study, it should be homozygous in nature. Okay, so either if I'm saying monohybrid, dihybrid cross, so we'll be saying that it is monohybrid for my trait of interest. 25,000 gene or complete genome can never be homozygous until I'm going for like some biotechnological tool. Otherwise, it will be something is always going to be heterozygous. There are certain studies where we found that even in case of rice, 34 years after that, still some genetic regions are kept heterozygous by nature. Nature never want equilibrium. Nature believes in disequilibrium. Okay, best example is eye color. India, black. You go to other parts, it is different color. See, it code color, maternal effect, and maternal like these two are different things. Only thing is that if something is regulated by maternal, it is just one generation after it is expressing. That's it. So it's not like it is not being governed by the genes which is present in cytoplasm. It is governed by the nuclear gene. Only thing is that if you for Suresh, if you need to understand this, you have to be very clear with the fertilization part. The moment pollen comes, then double fertilization, triple fusion, and then in case of monocot, dicot, what is formed? So only embryo is formed because of fertilization. Rest other parts are formed because of mitosis. Okay. But next generation, this embryo, which is like only F1 in case of plant, once we show the seed, this plant becomes F1. No? So embryo is F1, rest everything is the previous generation. So the seed of that becomes F2. Okay. Again, embryo becomes F3. So that clarity is required. Okay. So maternal, like nothing like effect. Okay. It is governed by just one generation, like afterwards. So that's the reason it won't be interfering with Mendel's study. Hopefully, you will be clear. Otherwise, you need to just little bit need to go through this fertilization part. That how which part is forming what. Then it becomes clear that why seed coat color is one generation, not only seed or whatever is there, except for the embryo, everything is going one generation back. Seed coat color is included. It is mentioned also in the paper. And anything else?
Participants can unmute and ask questions.
You can ask.
Sir, there is one more question in chat box, sir. Sir,
Yeah, I could see the like 7 trait affected by transposable element. There are so many things are coming once each genome is being sequenced. That transposable elements are there, but the trait what Mendel studied, we don't know those lines. Okay, we are studying the trait, but what line he is having, what line I am doing. Okay, even till today, if you make the cross, if you are working somewhere, you have P with you, you'll be finding somewhere like 3:1 is going to be there if your population size is good. Okay, so it's not like that it is all are getting affected. And then Mendel has proved so many things, and then afterwards also many has worked, and they had also found that it is finding 3:1, although not exactly the kind of ratio what Mendel has. That's the only thing. Okay. Okay. In case of cross-pointed, okay, I missed while saying cross-pointed. No? Cross-pointed, if you want it to work, just need to be very careful that if you want to study genetics, okay, for that, we have to make it self-pollinated. So inbreed, like you need to go through like selfing, and then you need to derive inbred lines. Then only you can do any kind of genetics. If things are not homozygous in nature, you cannot do genetics. That's a very, very simple logic. Okay, so you may be able to develop some variety, something, but for understanding genetics, it has to be selfing is required, and inbred lines are being used to do this kind of thing. Some residual heterozygosity is always there. Okay, not only for cross-pointed residual heterozygosity, we generally use it for self-pointed crop. They used to maintain some regions are like kept heterozygous in nature. So only like nature can decide that why it is keeping certain genome as heterozygous, even if you are doing again and again selfing. Okay, even by the sequencing has also proved that it is any plant is never ever 100% homozygous. Okay, and then what made Hugo de Veres, Carl Coran, Sherlock to discover Mendel? No. They by the time 1900 came, everything was known. So they were working independently. I have shown a different country, different system, and chromosomes were known, microscopes were better, fertilization is known. So by chance that all three came with this understanding that this is how traits are moving, and they are sitting on chromosome, and then these are we can understand the things. And by chance this all happened in 1 year, that is 1900, and they never wanted to give credit to Mendel. They wanted to be like they credit themselves. But fortunately, unfortunately, all three in the same year from different country, by that time then they were forced to say that okay, it is Mendel which is the main person, not you are. But still, they got this credit of rediscovery. That's the only thing. It's not like accidentally. They were working on different organisms, and since they all proposed together in 1 year, so this rediscovery credit was given. So they became also immortal along with Mendel, just because of this discovery. Won't be finding any other example where rediscovery term is there. Okay? Because none of the scientists are so much ahead of his time. Darwin reportedly came across Mendel paper. Yeah, many things were written. Okay, many things were written. If you see, and then best part is actually so many stories are there in case of Mendel and Darwin. So Mendel was fully aware of Darwin. Mendel was fully aware of Nageli, but Darwin was not knowing who Mendel is. So he has sent his paper also. But after Darwin's death, it was found that it is unopened. Okay. So sometimes what we do now, whatever is written, that is final. We cannot change that now. So literature says like this, so many things were mentioned, but it is sure that Darwin has not read what Mendel has written, and that too Darwin was typical English person. He was not knowing, and he has sent his paper, which is in German. So even if it have open, I don't think that how to understand, no, because they are preoccupied with certain mind that this kind of theory is there. He is the proposer, and then suddenly statistics into genetics very difficult to accept. See, there are two terms, okay, like your equilibrium and disequilibrium. Two typical terms are there. So, what we understand in terms of first, we need to understand equilibrium. Then only we can understand LD. Okay. So, equilibrium means what? If your in a large random mating population. There are different terms, isn't it? Your mapping population has to be large, then random mating. Okay, random means what? Large means what? We come to this. And then if there is no mutation, migration, random genetic drift. So in nature, nothing like this. Okay. If you take example of humans, plants, anything. So what we mean by large? UP, Bihar, India, world, what is large? We don't know. Okay, how to define? Then random mating, not allowed at all. Okay, whether it is plant system or any system, we are not allowing random mating because US there is different, here it is different, then different types of boundaries were there. So if this is happening, then only your gene and genetic frequency remains constant. But making it large, we don't know. And then population or nature always keeps the thing in disequilibrium. And because of this disequilibrium, it is easy for us to map. Okay, if everybody is having black eye, you cannot map. So different eye colors were there. Just because of disequilibrium, then only we are able to like map the things, to understand the things. And you take one example of COVID. Okay, So COVID, if suppose everything is in equilibrium, so nobody could have survived. Okay, since disequilibrium was there in same family, there must be something, some interaction between the viral gene and human gene has happened. Okay, so one member is not survived, other only 101-degree temperature, another family member, sometimes even white to the children. So things are very, very like nature by default keeps the things in discipline, which is good for like survival of any crop, any species, whether it is plant or animal or human. Right. So no, see, there are two things, Jivas and this is altogether different. In Jivas, nothing like that. Okay, Jivas we can take anything. So they are maintaining homozygosity. I don't think that it is that much important. Okay, so Jivas we do go for the complete whole genome sequencing. Okay, so if you are it all depends what kind of mapping population I want. If you want very precisely one trait to be mapped, okay, so make the cross, go with F2, then RIL, what kind of nature of gene is there. If your gene is like major QTL, then things are going to be different. If it is minor QTL, things are going to be different. So based on that, we derive the cross. Sometimes we are very easy to map even F2 if your trait is that. Okay, so cross-monografic means we can maintain in the line, but no, Jivas, no need of like inbred. You can have even any population can be taken and then it can be used for Jivas. Only thing is that you must be knowing what is the size of your genome. Accordingly, we generate the data for Jivas. Okay. So nowadays, Jivas is going with WGRS. WGRS is like whole genome resequencing. So if my genome size, for example, 3 GB, so I'll be generating 30 GB at least for one genotype. Then your Jivas is going to be very different. Earlier, we used to generate your GB around 1 GB for 3 GB, 4 GB data. So their mapping is different. Okay. So accordingly, we change. So their strategy is different if we'll be going with Jivas. If you're going with single trait or two trait mapping based on the crossing, then homozygosity. Otherwise, not defined. How independent things. It's not like that one day they have like Arun, it's not like that one day they have discovered. It's a complete year by process. I have found, then I said that this is something happening in different part of world. And then it took years to say that no, it is not you, then Mendel is there. Okay. Not like that. Today, Modi ji is going to join as PM. So they are independently working. They have published it. They wanted to take the credit. Then afterward, it was decided, though no, it's not like that. Mendel has already discovered. And then all they three again went through, and it was said that they all are knowing also Mendel's work. So it's not like that that one day they rediscovered. No? It was in that year they have published, and then in due course of time, credit was given. Because see, once science has developed, okay, mitosis, meiosis, chromosomes, known, good microscopes are there, then it won't take much time to rediscover. Okay, there could be a reason that they all three might have read this paper and they just use different system. Out of these three, one has used even P also as a system. He might be knowing this. Okay, just wanted to prove. Fisher's conclusion was very simple. There was a paper. Google it and read it. He said that Mendel's data was not original. That's it. Okay. And he said that because he was a typical statistician. He said that the kind of closeness, that's the reason I showed, of data, what Mendel has reported, that kind of closeness of chi-square value is generally not possible in any biological system. Okay. So he has taken like 10 different cross combinations and he could not be able to reach to that. Doesn't matter. Mendel was very, very clear. So it was it was called like Fisher-Mendel controversy. So that paper is there. Interesting to learn without any conclusion. Don't judge. We are not here to judge. Just read it and enjoy what Mendel has found, what Fisher has said, what was the Fisher's logic, what was the Mendel's logic. Fisher is doing in 1930s. Mendel has already done way back in 1865. He did work for 10 years. We don't know how many crosses, how many plants he did, and what ultimately he has taken. He is not going to get any medal for his work. Na? So why he'll be changing any data? Nobody is otherwise also understanding what he's saying. Okay, and then he got engaged with his own work. So questioning is very easy. So I don't question anything in science. I just enjoy. So just because of Fisher controversy, he is also immortal. Huh? So sometimes we get just by blaming others, we can do. But going to that era, working for 10 years, doing independently, making notes, reading, publishing, sending it to like hundreds of person, and then before his death saying that one day one person, the science is going to be understood by others. That's remarkable. No question about it. Right?
Thank you, sir, for answering these questions. In the future, we are planning to conduct this kind of lecture every month. Those who are passionate about this field can make use of this opportunity. And I would like to thank once again Dr. Gyan Prakash Mishra, sir, for accepting our invitation and delivering such a inspiring, thought-provoking presentation. Thank you, sir.
Nice to
And
be in
to
touch
all
with you.
to all our enthusiastic participants, thank you for being part of this special occasion and for your active engagement throughout. Let us all continue to foster curiosity, promote collaboration, and celebrate continuing spirit of scientific discovery that Mendel so beautifully embodied. Thank you once again, and have a wonderful day ahead.
Nice meeting you all. Thank you. Thank you, Shiva, and all.
Thank
Bye.
you, sir. Thank you so much. Bye.